Dr. Bob Ganz. We're going to talk today about fiber and the microbiome, and also how the fiber and the microbiome, the human microbiome, interact and play off against GI symptoms and GI and systemic disease today. Super interesting topic. Fiber is all the rage, so let's get into it. And just in way of disclosures, I am a consultant to Minnetonka Research Labs, the manufacturer of one of the products we're going to be talking about today. So what is fiber? We talk about fiber all the time. What is fiber? So it turns out fiber is the edible, we can eat it, but non-digestible, we can't digest parts of plants that are composed of complex carbohydrates. So all fibers are complex sugars, greater than 10 carbohydrate monomers. We can eat the fiber, but we can't digest the fiber. So it turns out that fibers are complex carbohydrates that humans cannot digest. We don't have the enzyme systems to digest complex carbohydrates. Humans can digest simple sugars, glucose, fructose, lactose, sucrose, etc., but we cannot digest complex carbohydrates. Those are fibers. So all dietary fiber comes from plants. Fruits are parts of plants. So fiber, complex carbohydrates that come from plants, edible but non-digestible. Here's an example of a fiber. This is resistant starch or wheat dextrin, just showing the complex carbohydrate nature of fiber. Now, what is typical fiber in a Western diet? If you look at a typical Western diet, and we have all kinds of fibers here, you can see in the darker blue that's cellulose, and in the darker yellow hemicellulose. So about 50% of the fiber that we consume in a Western diet is either cellulose or hemicellulose. And the rest of the fibers that we ingest some lignans, beta-glucans, pectins, fructans, galacto-oligosaccharides, resistant starches, and gums. But at least half of the fiber in a typical Western diet is cellulose and hemicellulose. Now, we said that fibers are not digestible by humans. We can't break fiber down, but fibers can be fermented by bacteria. So humans, when we derive sustenance or nutrients from food that we eat, we call that process digestion. But bacteria, who obviously don't have a GI tract, when bacteria derive nutrients from foodstuffs that bacteria get exposed to, we call that process fermentation. So humans digest, bacteria ferment. And when bacteria get exposed to fiber, the fiber that bacteria get exposed to can either be minimally fermentable, partially fermentable, or completely fermentable, depending on the kind of fiber that is ingested. Now, fibers, when broken down by bacteria, the bacteria create all kinds of metabolites and byproducts, but possibly the most important thing that bacteria create from fiber are short-chain fatty acids. And depending on the fiber that you eat and the kind of microbiome that you have will depend on the short-chain fatty acid profile generated by the bacterial fermentation of fiber. So for example, if you want acetate, which is shown in the green in this bar, pectin typically leads to more acetate in a short-chain fatty acid profile. And if you want, say, more propionate, which is in the yellow, that would be more wheat dextrin. And if you want more butyrate, that would be fructans and guar gums. So there are three main short-chain fatty acids that are produced by bacterial action on fiber. That's butyrate, acetate, and propionate. And depending on the fiber content in your diet, the kind of microbiota that you have in your system will depend on what short-chain fatty acid profile that you generate. So it's very interesting interplay between fiber intake and bacteria. Now, we said that all fibers are non-digestible, and fibers can be characterized by three different characteristics: solubility, fermentability, and viscosity bulking. So in terms of solubility, fibers can be soluble, partially soluble, or insoluble, meaning they don't dissolve in water. Fermentability we've talked about. They could be non-fermentable, partially fermentable, minimally fermentable, or completely fermentable. And then in terms of viscosity and bulking, fibers that are insoluble tend to be more bulking, but certain fibers can be partially soluble, that they can be gel-like or viscous, etc. So those are the three ways you should think about fiber: how soluble is the fiber, how fermentable is the fiber, and is the fiber completely dissolvable, viscous, or bulking, or gel-like. So categorizing fiber by characteristics, for example, if we look at psyllium, which is a common OTC fiber supplement, psyllium is partially soluble, minimally fermentable, and gel forming. And cellulose, hemicellulose, for example, is non-fermentable and insoluble and bulking. Wheat dextrin is almost completely soluble, almost completely fermentable, and non-viscous, etc. And other soluble and almost completely fermentable fibers are inulins, beta-glucans, pectins, guars, flaxes, acacia seed, etc. So acacia fiber, etc. So different fibers get fermented and metabolized different ways, and they create different characteristics in the body. So when you categorize fibers by various characteristics, if you have, for example, traditionally we've thought of insoluble fibers and bulking fibers as being good for constipation because they draw water into the colon and they bulk stool. And we've thought of soluble fibers, things like wheat dextrin, resistant starches, etc., as being good for gut health because those fibers are completely fermentable and they brew up a diverse microbiome, increase short-chain fatty acid production by their fermentability, decrease gut inflammation, etc. It's not that clean, however, because even the totally fermentable and totally soluble fibers can also sometimes improve constipation by changing up the gut microbiota and creating different metabolic characteristics. So it's not as clean as to say insoluble fiber for constipation and soluble fiber for gut health. It actually, there's a lot of crossover between these fiber characteristics, and it also depends on your gut microbiome. For example, just as an example of how complicated it's getting, there is an archaea, not bacteria, but there's an archaea that lives in the gut, part of the gut microbiome, called Methanobrevibacter smithii, or M. smithii. And this archaea produces methane, and methane slows gut transit. So about 30% of people with constipation have overgrowth of M. smithii. And we know that altering or overgrowing this organism with a robust, diverse microbiome can help this population of patients. So if you give somebody a soluble fiber, if they have M. smithii, that soluble fiber grows up a more diverse, robust microbiome, can overgrow the M. smithii, and can help constipation in that way. So again, not as clean as to say insoluble fiber treats constipation, soluble fiber does different things. Soluble fiber can also help constipation as well. And the microbiome characteristics can also determine gut transit as well as gut health. So that is sort of a long-winded way of saying why soluble and non-bulking fibers can sometimes be beneficial for patients with constipation or irritable bowel syndrome predominant, or constipation predominant irritable bowel syndrome. Now, let's switch our attention to the microbiome. So the human microbiome is immense. There's about 100 trillion bacteria that live in our system, mostly in the colon, although the entire GI tract has a unique microbiome. So the esophagus has a microbiome, which is actually regional. The stomach has a microbiome, the small intestine has a microbiome, etc. But the colon carries the most immense amount of bacteria, about 100 trillion bacteria that live in the colon. And the human gut is dominated by 4 main phyla of bacterial types. So Firmicutes and Bacteroidetes are the 2 gram-negatives that dominate the human gut. Acintobacter and Proteobacter are the 2 gram-positives that dominate the human gut. These 4 phyla account for 90% of account for most of our gut microbiota, with Bacteroidetes and Firmicutes representing about 90% of the gut microbiome in humans. And these 4 phyla are preserved actually in mammals. So it's a very similar picture for other mammals, including dogs, cats, mice, rats, etc. Mammals tend to share a lot of similarities in terms of their gut microbiota. Now, when you have a gut microbiota, like humans do and other mammals do, and you expose that gut microbiota to fibers that can be fermented, then that gut microbiota then creates all kinds of interesting products and metabolites. We've already talked about short-chain fatty acids, but the microbiota also convert primary bile acids to secondary bile acids. The microbiota creates all kinds of immunomodulators and cannabinoids and all kinds of interesting things. So there's this complex interplay between fiber and the microbiota, as we've said before, that determines what kind of metabolites are produced, and this will help modulate gut health. So in health, when you have a very diverse, robust fiber intake, and you have a very diverse, robust microbiome, then that interplay between fiber fermentation and the microbiota creates all kinds of healthy metabolites that then interact with the colonocyte receptors or the small intestinal receptors and create all kinds of good things that happen in your GI tract in terms of the proper mucus production and tight cell junctions, etc. So a very complex interplay between fiber, the microbiota, and then the gut lining and the gut receptors determining gut health. So as an oversimplification, in a healthy situation, you have the right fiber intake, the right gut microbiota, diverse, robust gut microbiota. This creates a very robust and healthy gut lining with proper mucus production and tight cell junctions, which leads to proper energy intake and proper blood glucose dynamics, blood glucose absorption, etc. In an unhealthy situation, shown on the right of this slide, then you either have the wrong fiber intake or you have a dysbiosis of the gut microbiome, and that faulty interplay between the wrong kind of fiber intake or a paucity of fiber intake and the wrong kind of microbiome or dysbiosis creates an inflammatory microbiome. You don't get proper mucus secretion or mucus production, and you can get inflammation in the gut lining from this complex dysbiotic interplay, and you can get cell-cell separation, a so-called leaky gut, if you will, where metabolites and inflammatory mediators leak through the intestinal lining and create all kinds of secondary downstream effects. It can affect energy intake and can alter healthy blood glucose and insulin dynamics into an unhealthy situation. And so this is an oversimplification, but it kind of gets the idea across of how these things interact to create either a healthy gut epithelium lining or unhealthy epithelial lining. Now, what happens to the microbiome as we age? As we age, we get decreased muscle mass and decreased subcutaneous fat, which is why people get wrinkles. And as we age, our muscles start to atrophy. When this happens as we age, invariably we get a reduced diversity and a reduced number of gut microbiota. So the number of microbes decreases in our GI tract, and the range of species declines. It's a very consistent finding in aging studies. As we lose skeletal muscle and as we lose subcutaneous fat, our microbiota get impacted in a deleterious fashion. So we lose beneficial non-inflammatory genera and species like Bifidobacterium, Faecalibacterium, Akkermansia. Those decrease as we age, and we get an increase in pro-inflammatory genera and species like Clostridium, Enterobacter. Those species increase. We get decreased or altered short-chain fatty acid production as that happens. We get altered bile metabolism as that happens, and we can get impaired vitamin synthesis. So for example, vitamin B12 synthesis can decrease, which is why a lot of people become B12 deficient as we age because of this altered gut microbiota. And we also get gut barrier dysfunction with transit of lipopolysaccharide and other inflammatory mediators across the cell wall. And so as we age, we tend to get more local gut inflammation, more systemic inflammation, and this can impact glucose and insulin dynamics. We get relatively insulin resistant. Glucose tends to go up as we age, all because, well, I shouldn't say all because, but at least in part due to this alteration of microbiota as we age. And there can be also impact on cognition. Remember, there's an information highway that goes between the GI tract and the brain through the vagal nerve and other signaling pathways. And so an altered microbiota can impact cognition and lead to things like mood swings, sleep disturbances, possibly even things like Parkinson's and Alzheimer's. Similar effect to the microbiome occurs during menopause. So during menopause, women get reduced estrogen and progesterone, and both of these hormones interact with the gut bacteria. And so you get a reduction in bacteria that because of decrease in estrogen and progesterone, and you also get a reduction in bacteria that can metabolize estrogen. So you get decreased estrogen absorption. This leads in women, at least in part, this effect in part causes increased visceral fat in postmenopausal women or perimenopausal women, osteoporosis and insulin resistance, similar to a general aging, but it's a little bit exaggerated in women during menopause. You get a decrease in vaginal lactobacillus, increase in vaginal E. coli and anaerobes. So this can lead to more urinary tract infections. You get a loss of estrogen-mediated vagal nerve signaling with altered brain-gut axis pathways. And so postmenopausal women can get mood swings or worsening moods, cognition issues, and altered sleep patterns. So that can also happen during menopause. Diet, exercise, and fiber supplementation is key then for women going through perimenopause or menopause. Now, trying to wrap this up, there is no perfect fiber. No single fiber covers the entire spectrum of bulking, viscosity, solubility, and fermentation. So fiber combinations have added value. The more diverse your fiber intake, the more diverse your microbiome will be, the greater numbers of microbiota you'll have, and the better effect you'll have on your GI tract and hopefully systemic circulation. So various fibers with various characteristics yield different clinical results. We've already talked about that. But in general, fibers with multiple properties or combination of fibers or fiber blends yield the most microbial diversity and the healthiest microbiome. So there is a product that I consulted on called Relievance. We helped develop Relievance Fiber. This is a new fiber supplement, and Relievance Fiber is a blend of 8 natural fibers. So it contains psyllium, wheat dextrin, beta-glucan, inulin, pectin, guar, cellulose, and flaxseed. And this 8 combination fiber is a nice mix of soluble, insoluble, fermentable, partially fermentable, and viscous, non-viscous. And this fiber combination is very, very healthy in terms of creating a diverse, robust microbiome. So Relievance is 8 fibers in 1 dose. This is just a pure fiber product. There's no sugar, no additives, no preservatives whatsoever. This is just 100% fiber, and it's tasteless. And Relievance is predicated on being just a pure fiber product with nothing added to it at all. So a novel fiber product, it's an 8 fiber blend. It's a very novel combination and a relatively low amount of fiber, but each fiber has various characteristics of solubility, fermentability, and viscosity. And this product helps to restore gut health by restoring a healthy, diverse gut microbiome, helping to decrease gut inflammation, and helps to restore a normal bowel habit and improves bloating, gas, and digestive discomfort. Relievance has been studied. This is a study of Relievance patients with gas, bloating, and irregular bowel habits were assessed, and they were assessed via Rome criteria to get into the study. But in terms of gas and bloating assessment, after 6 weeks of Relievance, gas and bloating decreased by about 50% in the patients on Relievance. And in terms of improved bowel habit, there's about an 88% improvement in bowel habit, where patients on this product went from 1.7 bowel movements a day to, 1.7 bowel movements a week up to 3.2 bowel movements a week on this product. It's an uncontrolled study, but the effects were pretty impressive in an uncontrolled study. And controlled studies are being performed to try to confirm this, along with some microbiota analysis and 16S RNA subunit typing to see if we can tease out the changes in the microbiota that lead to these good effects. And I'm going to end it there, but hopefully that's a good, quick primer on fiber, its impact on the microbiota, and impact on gut health and systemic health as we age and as we go through various things such as menopause, et cetera. Thank you for your attention.